afternoon, everyone. Thanks the organizing committee and Professor Lee for inviting me to this important topic. Today, my topic is emerging issues after pneumococcal conjugate vaccination program. Undoubtedly, pneumococcal conjugate vaccine have substantially reduced the incidence of invasive pneumococcal disease caused by vaccine serotypes. However, replacement disease with non-vaccine serotypes remain a concern. Furthermore, these non-vaccine serotypes are increasingly exposed to systemic antibiotics, potentially enhancing the rate and extent of antibiotic non-susceptibility. Serotype replacement varied geographically. Striking differences have emerged between the European country and the United States. In the United States, the incidence of non-vaccine serotype invasive pneumococcal disease in children has been stable after the PCV13 introduction. In the elderly, the incidence of a non-vaccine serotype invasive pneumococcal disease has been stable as well. Impacts of serotype replacement hasn't been reported in the United States. Taiwan's situation resembled that in the United States. The national catch-up primary vaccination schedule successfully reduced the incidence of IPD in children by almost 17% reduction. A modest herd effect was also observed with a 37% reduction in the incidence of IPD in the elderly. Serogroup 15, including 50 ABC, replaced serotype 19A to be the leading IPD serotypes after 2015. On the basis of national surveillance data, serogroup 15 and serotype 23A were the most common non-vaccine serotypes, both among children and the elderly. Other countries had quite different situations. England and Wales reported 37% lower incidence of invasive pneumococcal disease since the period before PCV7 introduction, but non-vaccine serotype accelerated since 2013. Increases in non-vaccine serotypes have negated reductions in vaccine serotype disease. If we look at the age-specific incidence, in young children, the incidence of invasive pneumococcal disease remained decreased despite of little increase in non-vaccine serotype. But there was little or no reduction in the incidence of IPD in the elderly owing to an increase in non-vaccine serotypes. Serotype 8, 12F, and 9N were responsible for more than 40% of invasive pneumococcal diseases by 2016. Serotype 50A, 22F, and 33F mainly increased in the adults. In French, as expected, after PCV13 introduction, The incidence of IPD decreased to a nadir in 2015, but a rebound in cases both in children and adults in 2016 and 2017, driven by several non-vaccine serotypes, jeopardize the long-term PCV benefits. The main serotype involved in the increase was serotype 24F in young children. The main serotype involved in the increase in the elderly were serotype 22F, serotype 8, 9N, and 12F. Factors for differences in serotype replacement include surveillance practices, transmission dynamics, population risk factors, and pathogen evolution. Regarding pathogen evolution, there are two mechanisms of serotype replacement. One is expansion of non-vaccine type lineage. It is a new non-vaccine serotype lineage with a new non-vaccine serotype. It emerges after PCV introduction. It's just because those vaccine serotypes decreased. The other is within lineage expansion of non-vaccine serotypes. It is a pre-existing vaccine serotype lineage with vaccine serotype. Under the selective pressure of pneumococcal conjugate vaccine, the old pre-existing lineage shift its serotype to a new non-vaccine serotype, but inside they remain the same. They were the same lineage. For example, serotype 35B emerged after the introduction of PCV13. The increase was caused primarily by serotype 558 clone. Serotype 35B sequence type 558 was rarely detected before the use of PCV. But during 2015 to 2016, another lineage, sequence type 156, increased. Serotype 35B, sequence type 156, was a capsular variant of a previously famous clone, serotype 9V, sequence type 156. After the use of pneumococcal conjugate vaccine, this old pre-existing lineage shifted its serotype to non-vaccine serotype 35B for persistent circulation in the community. The spread of this particular clone is of a concern in view of past predominance of the pathogenic sequence type 156. Let's look at the Taiwan's data. During 2013 to 2017, at Chang'an Memorial Hospital, we collected 206 invasive pneumococcal isolates. Serotype 50A, 50B, and C significantly increased during the study period. In penicillin and meropenem non-susceptibility test, serotype 50A, B, C were the most common penicillin and meropenem resistant clone. If we look at this serotype BC strains by whole genome sequencing, we can find most of them belong to sequence type 83. Sequence type 83 was a single locus variant of sequence type 81. Before the use of PCV, sequence type 81 mainly expressed serotype 23F. After the use of pneumococcal conjugate vaccine, this clone shifted its serotype to 50BC and made recombination at other genomic site to be sequence type 83. This specific clone, serotype 20F, sequence type 81, was very well known for its multi-antibiotic resistance and genomic plasticity. By prediction analysis of recombination site, we can see serotype 50BC sequence type 81 had 12 recombination sites compared to its ancestor serotype 23F sequence type 81, including the capsular polysaccharide region. This is why it shifted to serotype 50BC, and the loci called SPI, it made sequence type 81 to be sequence type 83. And there were other recombination sites at their genome. On the other hand, serotype 50A also increased during the study period. Most of them belong to sequence type 63. Remember, they were also penicillin and the meropenem-resistant clone. The specific meropenem-resistant serotype 50A sequence type 63 clone has been reported to be increasing in Japan. By whole genome sequencing, Japan study found the specific meropenem-resistant serotype 50A sequence type 63 clone was different from those sequence type 63 clone in the United States, UK, and Canada. The meropenem clone was involved from a meropenem susceptible 50A sequence type 63 clone. The meropenem susceptible 50A sequence type 63 clone in Japan and in Taiwan acquired a PBB1A from a previous wild-spreading 19A sequence type 320 clone and generated a novel PBB2B to be meropenem-resistant 50A sequence type 63 clone in Japan and Taiwan. In our transformation study, we use R6. R6 is a serotype 2 strain without capsule, and we transform the novel PBB2B into R6 strain. And we found the novel PBB2B involved in increased meropenem resistance. We can see the wild-type allele R6 had meropenem MIC 0.015, and when we gave the R6 novel PBB2B, the meropenem MIC increased to 0.06. And also we found the 1A from 19A sequence type 320 was associated with breakthrough meropenem non-susceptibility. The meropenem MIC increased to 0.5. Conclusion. It's our conclusion. Not all pneumococcal strains are equally frequent colonized at the nasopharynx. We encountered the next generation colonizer. They may generate new antibiotic resistance genes. Previously successful clone may change their capsular type or serve as a gene pool reservoir and donate antibiotic resistance genes. So I think reducing the consumption of antibiotic by judicious use of antibiotic remain very important in the future. Above is my report. Thanks for your attention.